Hi Arushi. Hello Arushi.
Hi, ma'am.
Hello. Arushi, you had one question na, in the discussion forum. You posted two questions, I guess, on
I
mapping.
don't remember, ma'am. In the mail, ma'am.
Yeah, in the mail. Yes.
Yes, ma'am.
So I guess in my last live session, I've discussed one of them. So you can go to my YouTube channel and just have a look. Okay.
Okay, ma'am. Thank you.
Okay. So I'm not waiting for anyone else as nobody else is there. I'll just straight away start with today's discussion. So we'll be discussing week six contents, and as I start sharing, just give me. Okay. So if you have any doubts, just interrupt me. And also, as you know, we'll have three sessions, 40 minutes each. One session gets over, kindly leave and join in a minute. Okay. So,
Okay, ma'am.
okay. Just confirm if my screen is visible or not. Yes. Hmm. Yes. And if my screen is moving or not, confirm that also. It is right?
I'm moving, ma'am. Yes,
Moving, no?
ma'am.
Okay. Fine, perfect. So, okay. So mostly we'll be discussing about basics on cell cycle, a bit of dev bio, and mostly cloning. And so we'll be discussing these topics very briefly and try to solve questions that were in the sixth week assignment. Okay. Okay. So before solving questions, let us briefly discuss like what cell cycle is, what are the different phases of cell cycle, and what happens in the interphase in mitosis and like that. So as we know that there are few phases which are known as, so why cell cycle is required? It is required so that the cells divide and its nuclear content, its cytoplasm, everything divides into two, into daughter cells, and it keeps on continuing. Like ultimately, the idea is or the aim is to have more cells with the same genetic constituents. So the division mechanism is what we're going to discuss here. And so specifically mitosis has majorly majorly four phases. One is the prophase, one is then the metaphase, anaphase, and telophase. And in between, there are like early prophase, late prophase, early metaphase, pro metaphase, early anaphase, like that, and late anaphase. So, if we just one by one discuss what these are, so what happens is your entire cell will divide into two and and the contents of the cell will get equally distributed into two cells. So in order to do that, hmm, so in order to do that, what happens is the internal things in the cell, the nucleus, the membrane, all of these, the nuclear membrane, the centrioles, they have the mechanism by which it prepares the cell so that it can divide into two. So if a DNA or the genetic constituent or the main the genetic power hub, as we say, has to get divided, so the nucleus has to disintegrate and it has to break. So what will happen is first, the very first one in the early prophase, what happens? Hmm, hmm. So what happens is the chromosome, the centrioles have to replicate before the end of the interphase. Centriole pairs like they start moving apart so that ultimately they can form this kinetochore complex. And as a result, the chromosome that you see, what is inside here, this part, okay, the chromosomes, they shorten, they thicken, and they become condensed so that they finally can take a shape like this. We are very aware of this shape of chromosome, right? this is like the not the condensed one, it's the elongated one. It's not, it's less condensed one. So that is how the chromosome is now. But now it has to divide, it has to get separated and equally distributed. So it starts condensing so that it can reach up to like this. So in the early prophase, the cell starts like this. The centriole, like if you see, this is one cell, it has just one centriole and then the nuclear region. So this centriole becomes two. So it becomes a pair. And these, the inner part in the nuclear, like within the nucleus, they start condensing. And also nucleolus, that is this organ, sorry, this organelle, this one starts disappearing. And the nuclear membrane starts slowly disintegrating, because if the nuclear membrane doesn't disintegrate, how will the chromosomes come? Okay, so that is all. So what happens in the late prophase? The mitotic spindles start appearing, like from the centrioles. If this is the centriole one, like this, and it's a barrel shaped one. I'm not, yes. So like this, the spindle fibers start generating from here. And now still it's outside the nucleus. The nucleus is not completely disintegrated, so still it's within the nucleus. So now each duplicated chromosome becomes visible at two sister chromatids. That now the chromosome has completely condensed and it has become in this form. In this form. So now you see this is visible. These two sister chromatids are visible. And also the nuclear uh, envelope breaks down near the end of prophase. Like at the end of prophase, it will start breaking down. As I mentioned, that it the process has just start of breaking down. Now coming to, so the prophase is complete, early prophase and late prophase. So generally in class 12 or in during our college, we used to read only prophase. Like this discrimination between early and late was not there. So actually this is not a single event, it's a eventual event. It happens, it's a eventual event in a continuation. It's not like just suddenly everything decondenses, the chromosomes are visible, the centrioles have started producing like the mitotic spindle. No, that doesn't happen, because it's an eventual process. So the early and the late phase, that is why the discrimination is there. And okay, now coming to the pro-metaphase. So in metaphase, the idea is that all the fibers will expand and will bind to the centromere region of the chromosome. So centromere region is this one. If this is a chromosome, here is the centromere. To this, it has to bind. Now, why? This will bind and they will all align like this in one plane. And this plane is known as the equatorial plane. Why? Because, I'll just take one more. Because the division will happen right in between this plane. So all the chromosomes will align along the equator. And then what will happen? Then these kind, now the kinetochore is also attached to it. So that marks the metaphase. So the what happens in the pro-metaphase? The spindle enters the towards the nuclear area. Now this was former nuclear area, because initially this was the nuclear area and it got disintegrated. The nuclear membrane, everything came out and it's now condensed. So the kinetochore attached to the sister chromatids were in the centromere region. Now you see in this picture, there are many non-kinetochore microtubules and kinetochore microtubules. So this non-kinetochore microtubule, as you see, they are not attached to the centromeric region or the sister chromatid anywhere. It is just random. So these are the non-kinetochore. Okay. And then in like what happens in the metaphase, the kinetochore in the next phase is like the kinetochore microtubule orients duplicated chromosomes on the metaphasic plate. Like what happens is, if one kinetochore is attaching to this, I'm sorry, to this part, the other one will attach to this part. So there's a tension and it can be pulled apart like this. So that is why the kinetochores or the spindle fibers attached to the orientation is generally opposite. Like from one centriole, if this is one centriole, one will come like this, and the second centriole from like this. Okay. And that is how the kinetochore attaches and it creates a tension and it pulls. So once it pulls, that marks the beginning of the anaphase. So what happens? The two centromeres of each pair of sister chromatids separate and the resulting daughter chromosomes begin to migrate to the opposite poles. Like what poles? The poles means that towards the centriole, like wherever the centriole are, there the pole. And then marks the beginning of the late anaphase in which the two sets of now the daughter chromosomes or the parental chromosomes are equally distributed, so they can easily form the daughter cells now. So what happens is the two sets of daughter chromosomes approach the poles. Cytokinesis division usually begins, like the main division of the cytoplasm, cytokinesis of the cell. Also, so the cell division begins, in which they are equally distributed and they form two different entities or two different cells. So that means the division has reached to the telophase. So telophase means again the nuclear envelope will regenerate or again reform. Chromosomes again will become extended and less visible like this, in a very coiled manner, and also the nucleolus will reform. But the cytokinesis keeps continuing. Why? Because now still they are not completely separated. And in the interphase, then what happens in the interphase? That the cytokinesis is complete and chromosomes are expanded and are no more visible, like two different cells, and you can see how different these are. So this is the entire process of mitosis in an animal cell. Let's try to solve a question regarding this. Okay. So the question says that the cell cycle is a set of activities that take place inside cells to produce daughter cells by dividing the cell's cytoplasm and nucleus. The cell cycle has four key stages that the cell goes through. In any eukaryote, what is the ideal stage for counting the total number of chromosomes? And also at which point or what point do chromosomes align on the equator of spindle fibers? Pick the correct combination. So, any idea? Let's see the picture once again. Maybe if it's from here, you'll understand. So, what do you think is the best way to quantify the number of total chromosomes in cell cycle? And also at which phase does they all align at the equator? Now, the second one is simple and clear. Which phase it could be? We have the chat box. B. Okay. Yes, you're absolutely correct. B. Why B? Now let's see the picture once again. What is happening in metaphase? So this is metaphase. You can see clear chromosomes here. Not even that clear chromosomes you will find in the prophase. It's like I said, it's an initiation. So if you have to exactly count the number of chromosomes, it should be metaphase. And the second question was quite straightforward. Just looking at the picture, we can make out that only in metaphase all the chromosomes will align at the equator, like at the normal, like equatorial position. Okay. I hope I'm clear on this. Any other questions?
No, ma'am.
Okay. Okay. Now, before let's look at this question first, and then we'll come to the explanation part. Okay. So, suppose we are studying a cell division in a metastatic cancer cell line. So your senior gave you a subline of cells where she had fused kinetochore proteins with GFP. So what is a GFP? That produces a green fluorescence. So it's a fused protein. We were discussing in RDT, like when we were discussing RDT, I guess we had discussed that where do we use GFP tags and what is the necessity of using GFP tags. We had discussed this. So one is to easy isolation, like when we have to isolate our target in a heterogeneous cell population or in a heterogeneous protein complex mixture. So then we can have a tag to our target of interest. So, and then we can easily use any kind of chromatography or affinity protocol to isolate or elute our proteins. One is this, and the second one is visualization. Now, whenever you are tagging a fluorescence colored protein to it, so and it will give you a color when you are exciting it with a particular wavelength of light. So, like that, also when you observe the cells under microscope, if this protein is present in a specific organelle, that organelle you'll see the particular fluorescence color in that organelle. So, similarly, she did that she had fused the kinetochore proteins with what? With GFP tags. Okay. Now she grew the cells, and after reaching confluency, she harvested the cells and mounted them on a slab. So, in now what is confluency and what is mean by harvesting? So, if I just try to brief it in terms of cell culturing, animal cell culturing. So, reaching confluency means that in animal cell culturing, if you are aware, the cells are grown in a flask and they become adherent. So, the time or the part at the time taken for all the adherent cells to cover the entire surface is known as confluency. Now, generally, when we have to take cells for experiment or we need the cells which have already reached confluence, it's 80%. Like if 80% is, if that is covered, then we say, okay, it's confluent, we can now use. Sometimes it's 50, 60. That depends on the eye of the experimenter, that according to him or her, what is the confluency. And also, as the confluency increases, what happens is these are adherent cells, so they will start topping on one another, and then you will eventually they will die. So it's very important that we don't grow till 100% confluency. So similarly here, that is the concept of confluency. So confluency is actually a checkpoint that okay, my cells have grown, now I can use them for the experiment that I was planning. So now my cells have reached confluency and we harvest them. Now this harvesting is a very common term that is used in biology. So what it means that it is to use it for an experiment or to use your cells, right? And there's one more term that I would like to introduce you all to, that is passaging. Now, what happens is when we are doing cell culture, one is we harvest and do it, keep we continue our experiment, and one is we save it for the future. We keep on letting the culture grow and grow so that culture doesn't die and it is maintained. The life cycle is maintained. So that is called passaging. So here, what we are doing, we need it for the experiment, so we are going to harvest it. So after reaching confluency, you harvest the cells and mount it on a slide. So it's very clear that we are going to do a microscopy experiment on. So now the question is, which part of the cells do you think will show fluorescence? Now, if we harvest the cells at the logarithmic growth phase, followed by cell lysis and centrifugation, then what part of the solution will contain most of the green fluorescence? So first, let's break the question. Which part of cells do you think will show fluorescence? So let's go back to the explanation. I just have one slide. Okay. So what has your senior done? That your senior has taken your kinetochore protein and has tagged it with GFP. So when that protein is actually expressed, it will become green. Okay. And then uh, she or he has like that you have actually harvested it and then performed a cell lysis and a protein and an extraction protocol. And then you want to see two things. One is uh, which part of the cells will show you fluorescence when you're observing under a microscope, and what part of the solution, like in this experiment, what part of the solution will have your target. So, like which pellet will have the fluorescence. So just guess. Let's discuss what and why could be the reason. Okay. Any idea? Anyone? Is the question clear to all of you? Just answer in a yes or no using the chat box, that if the question is clear to you or not. I'll repeat the question once again and then maybe we'll discuss the answer. It could be a tricky question at this point when all these parameters like logarithmic growth phase and all of these come. So is the question clear to you? Okay, yes. So Arushi, what could be the answer and why? Okay, let's discuss. Let me discuss this. I guess it will be easier for you. So, as we discussed, what is kinetochore? So, this is the centromeric region, and this is the kinetochore portion. And from here the microtubules come and attach to itself, right? To the centromeric region where in the kinetochore. So, these are the kinetochore microtubules, right? Now, if we are fusing the kinetochore protein, so obviously in the centromeric region, my fluorescence will be observed because it will be attached to it. And this, when if this is a cell, but this is not a cell, then this part will be, I should have used a green color. So this part will be giving you a green signal. That means that centromeric region. Okay. And now when we are performing an extraction protocol, so what do we do in an extraction? What has she done rather? That she has done a lysis and a centrifugation. So, okay. So we take a cell, we give it like we try to lyse the cell wall with a specific reagents like urea, NP40, things like that. Maybe like mostly detergenic substances and salts. And then what will happen on subjecting the cells to a lysis buffer, it will lyse and the constituents will come out, like whatever is within the cell. Now everything is going to come out and then you're going to centrifuge. So what will happen in centrifuge with because of the heaviness or because with the according to the weight of the components, they will either settle down or float. Now cell debris, walls, these kinetochore or any kind of small organelles, they will settle down because they're heavier and the rest of the solution will have the proteins and the other lighter part of the cell. So what is the thing? So this the it will be the third, like, so it will flow. Now, as I mentioned, you settle down. So whatever settle down is known as the pellet. This is the pellet. Proteins, yes. You see, proteins are released into the cells. This is the pellet, like the cells remaining are centrifuged. Okay. Supernatant and proteins are saved. Okay, yes. So what happens is, I'll just draw it once again for you. So if this is your append of tube and you have, suppose this is your solution, all the debris will come in the pellet. And in the solution, you will have your other lighter components. So, according to this question, and because this centromeric region, it is part of an organelle, right? So, it will also settle down. So, you will find the fluorescence in the pellet. Here, you will have the fluorescence. You can see that. Yes. Yes. So, this is the answer. The tick mark went above. So, it is this one. That centromere only and pellet will contain most of the fluorescence. So is it clear why it's not else and why it's centromere? Now, if why not A or B? So A and B claim that both chromosomal ends or telomeres will have the fluorescence. Supernatant will contain majority of the green fluorescence. So that supernatant part is, I guess, clear that because this centromeric region is part of the chromosome and it's going to be heavy, so it'll settle down and it will be in the pellet. But why not the chromosomal ends? Chromosomal ends because chromosomal ends are not attached to kinetochore or the microtube. So because we have the kinetochore attached, the kinetochore is only present in the centromere region. That is where it gets attached. So that is the reason why it will be only centromere and not anything else, not any other location on the chromosome. Is it clear for all of you?
Yes, ma'am.
What happened to you, Arushi? You were quite interactive in the last sessions. Are these concepts very new to you?
Little new, ma'am.
Little new. So you can, if you want, we can, like, I can break it down more and like go more into basics if you want. So we can do that also. Okay.
Okay.
Okay. Is this clear? Like, are you clear of the why the supernatant doesn't have the fluorescence and why the pellet has it? Is this clear to you?
Yes, ma'am.
Okay. Okay. And for rest of, and I guess there are more two participants. Is it clear to you? Amardeep and Ozma? Yes. Clear. Okay, good. Very good. Now, now these are the events of the cell cycle. I guess we have discussed what happens in a cell cycle. So, and one more thing, what is the maximum number of times chromosome can replicate per cell cycle in human keratinocytes? So we'll discuss that also. Like there's a concept behind it, but before that, let's just see what is the order. Let's look at the picture and see that what is the order of the different what will come after what. So just try to guess and then we can discuss. Okay, let me help you through the process, I guess. So what did I say? First, the nuclear membrane has to disintegrate, or what will happen? The centrioles will get into different poles, right? So, okay, first what will happen? The these are not condensed, these are quite coiled in nature, the chromosomes. So which one will come first? Three?
Three, ma'am.
Three. Okay. Then you see the centrioles are migrating to the poles. Then it's two. And also the chromosomes have started. I have answer. Three. Okay. 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 You mean C? The answer is C. Okay. We'll just discuss and see. They have started like going to their poles, right? Then they're completely towards the poles, and the sister chromatids are this one, four. They're quite towards the poles, and they have, the sister chromatids are aligning towards the equator, right? Or these are, you see the nucleus is still intact. This one, four, if we see, sorry, five. So, sorry. Yes. So, it's just changing. So, yes. So, you see, the nuclear membrane is still intact, and they have completely gone towards the pole, the centrioles, but the sister chromatids are yet to be aligned to the poles, to the equator. So this marks the beginning of the prometaphase, right? And then the spindle fibers, this one, the spindle fibers have attached to the sister chromatids in the kinetochores in the opposite way, you see. And the last one is they have pulled them towards them in half, and then you see a cleavage is starting to happen. That is, the telophase will begin. So it is 3, 2, 5, 4, and 1. Clear?
Yes, ma'am.
Clear? Is this step and this step confusing you by any chance?
No, ma'am.
It's clear now. Here you see the astral fibers or the kinetochore or the microtubules are not there. So this is coming in the first, and because it's here in the fourth, it is it's a late metaphase or the end of the uh, metaphase. Okay, that is how it's happening now. There's something called the MDM helicase. That is the reason why replication is limited to just once. Like it's not that then one cell will have consistent, like they will keep on replicating. No, that doesn't happen. Per cell in one cycle, only one time or only once the replication can occur in per cycle in any cell. Okay. So so the maximum number of DNA replication is one. Hence the answer here is odd A, that is this order, and the maximum number of replications. Okay. Okay. So just let's discuss the next question, and then we can like the this will get over, and we'll move to the next segment. Okay. So the next question says that in a cell culture laboratory, you're looking at a glass slide that exhibits a certain phase of cell division. So if you are aware of it, I used to do practicals where they used to give me sections of cell and they used to ask us to identify which phase of cell it is. Has anyone done this practical? Anyone? Have you heard of this practical? Have you done it in person? Okay. I assume you have not. Yes. So you see a particular phase of cell division. So when you questioned your senior about the right phase, he or she said that it was anaphase. You attempted to match your slide to some of anaphase's important points. Now choose the correct statement about anaphase. So till now, as much as we have discussed uh, anaphase, what it could be like out of the four options. Just give it a try. Any one of you? So I have attached a picture also of the anaphase. See, yes, Arushi, see, good. Yes. So it says it's anaphase. Why? Because the homologous chromosomes segregate from each other, moving towards the pole because the microtubule depolymerization happens at nucleocapsid. That is anaphase. You see, what happens is now they the pull starts. They start pulling the sister chromatids, whatever they were attached to the kinetochores and all. So the homologous chromosomes they segregate. You see, this is the segregation. So they have started segregating from each other, move, and they move towards the pole because these microtubules they start depolymerizing. Where? From this region, from the kinetochore region. So automatically they start doing that, and so it disintegrates or moves. Is it clear? Uh,
Yes,
let's go
ma'am.
back to the first picture also. It will be more clear to you. Detailed one. Let's see here. So here you see they were attached to the sister chromatids. And what happened in the early anaphase, due to depolymerization event, they are now being pulled. As a result, finally they'll be pulled and made into new chromosomes. Okay. Okay. Now let's just shift to a bit of different, a little of cancer biology. We have okay. Okay. So rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo change. That is, by change, naming genetic mutations. This is the stage of transforming a cell that can be described best by which of the following option. Now, this stage of transforming cell. Now, which stage? Where the cells have now started appearing aberrant and disordered as they undergo the changes. Uncontrolled cell division, on the other hand, may result in overabundance of cell that appears normal under microscope. Now, there are two things to it. There are two parts of this question. One is that there's uncontrolled cell division that makes all the cell clump together and look like okay, they are normal under the microscope. Just number of cells have increased, which could happen due to any reason. Also, the cells have started to appear aberrant and disordered. So, what is this condition called? Any clue? Anyone? Let me just, I have kept a picture. So just have a look at the picture and see. what this picture means. This is the normal, this is hyperplasia, dysplasia, and this is cancer. Yes, metastasis. Okay. But where do you find the option metastasis? Okay. So, before developing cancer, we know that proliferation, uncontrolled proliferation leads to cancer. Okay. But apart from that, there are many other things also. Now, even before developing cancer, even if you're having that uncontrolled proliferation, it is not always necessary that it will lead to cancer. Okay. We have benign tumors also, apart from malignant tumors. So, in those cases, sometimes this proliferation or this way the cells appear, these are like reversible. So, what happens is, suppose this is my normal cells, this one. Okay. You see, this is the normal cell. When you are looking at the hyperplasia, what is happening is, if you see, number of cells have increased. Like the number of cells, the growth is much more than the normal. You see, each cell, the nucleus is also big. It seems big. It's not big. Actually, number of nucleuses have increased. That means number of cells have increased. Here, apart from the number of cells, you see, morphology and the size of the cells have also increased. Uh, is it understandable from this picture that the how the morphology is quite different in case of hyperplasia and dysplasia? In dysplasia, what is happening? Only the number of cells are increasing, like this uncontrollable proliferation. But in dysplasia, what is happening? Besides uncontrolled proliferation, morphology or the way the cells are looking, they look a bit disorganized, they look aberrant, right? That is also changing. Is it clear from the picture?
Yes,
I
ma'am.
hope yes. So you see, in dysplasia, this is happening. And in case of the fourth one, the cancer, this is infiltrating to the other parts. Or this is a basal lamina, it's infiltrating to the basal lamina and it's going above. Right. So, what does the question say? That rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo changes. This stage of transforming cells can be described best by which of the following options? Uncontrolled cell division, on the other hand, may result in overabundance of cells that appear normal under microscope. So, after this entire like discussion, what do you feel could be the answer? Like, what does these two things indicate? What, which stage? What does it indicate? Like the cells look appear normal, but they are actually overabundant in nature, but they will look normal, but they are not. D. D is hyperplasia, hyperextension. Um, what do you mean by hyperextension? You feel the structure is extended, that is why do you mean hyperextension? Is that what you're meaning by hyperextension? Look, there's nothing hyperextension. It's clearly written that uncontrollable cell division. So it is not extension, it is proliferation. So it will be something related to proliferation. That I'll give you a clue. It will be hyper proliferation only. But is it hyperplasia? Do you think so? That is it only what is saying that uncontrollable on the overabundant cell that appears normal under a Thank you, Amardeep, for joining back. Thank you all for joining back. I'll just share my screen once. Yes. So someone said that is hyperextension, hyperplasia. But what did I say? You have two conditions, and it's clearly mentioned that rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo changes. That is, it's over uncontrolled proliferation is there. Like definitely the number of cells are increasing, but also it is looking very aberrant under the microscope. So these two conditions, what could be the possible thing that a possible stage? It is not normal. Okay. So it is also not metamorphosis. Yes. C. Yeah. It's C. Why C? Because two things that if you look into this panel that this is from NIH, this definition is from NIH, that is in hyperplasia, there's an increase in number of cells in an organ or tissue that appear normal under microscope. So it's most of the time it's not detectable only that it's hyperplasia. So, and it's reversible, like it can go back to normal. It happens due to many reasons, it can again go back to normal. In case of dysplasia, the cells look abnormal under a microscope but are not cancer. But till now, they're not cancerous. So, when are they going to get cancerous? When they infiltrate into somewhere else. Like they cross the lamina. This is the basal lamina, and then they go to other places. So, that is where, or other places, I mean, other tissues. So, that is where it becomes a cancer. So these two, hyperplasia and dysplasia, are actually reversible, but cancer is not reversible. So it depends on the environmental factors and its environment, the environment of the tumor, that it will develop into cancer or not. So is it clear why it is dysplasia and why is it hyperplasia, and what are the different morphological changes that happens when a normal cell becomes a cancerous cell? So is it clear to all of you? Okay, let's see the next question. Okay, so this is a graphic representation of sequence of events that occur through the development of an embryo. So we need to select that the appropriate answers that the cycle is correct. Now each stage we have to define which is it. So any one of you? Okay, we have answer. C. Fertilization, blastulation, and gastrulation. So 2 and 3 both are blastulation. Arushi, I guess. Yes. Arushi? Is two can 2 and 3 both be blastulation? Anyone? No one. Okay. So what is embryogenesis? Or this is a process of development of an embryo. Okay, I have an answer. D. Yes. Okay. Let's discuss. So what happens is, uh, when an embryo meets, when a sperm meets an egg, they they join together and then fertilize. Okay, and they fertilize to form a fertilized embryo. So what happens is, then eventually it divides to form a complete fetus. So the first process, the first step in development of is fertilization. So by fertilization means the sperm meets the egg, and the sperm fertilizes, both of them have nucleus, and it fertilizes the egg, and they become the very primitive stage of an embryo, which is a two-stage cell stage. So that is known as cleavage. Why? Now cleavage means to form specific compartments or to form invagination, so that it forms different cells. So you see the two-stage step is the cleavage step, where they just started divide, followed by an stage, 8 cell state, which is known as the blastulation. So now the blastulation is a very important stage. Why? Because this actually decides that how the cell will be implanted. Now, the embryo needs to be implanted on the walls of the uterus, and then that is how the placenta will form, and the rest downstream process of development happens. So that is the blastulation step. What happens is, if you see here, it blastulation, it forms a cavity like thing. This is known as the blastocele, and these cells are the trophoblast cells. And what happens is, here you will find ICM, or the inner cell mass, which is again a very good source of embryonic stem cells, ESCs. Why am I focusing on ICM and ESC? Because this is something which is very important for animal biotechnology also, when we are discussing transgenic animals. Okay. So yes, now what will happen? Then it will again divide further to form the gastrula, or which is known as, the process is known as gastrulation, and then it divides into ectoderm, endoderm, and ecto, endo, and mesoderm. Okay, so that is the very brief this process. So this is the blastula, and this is the gastrula. The third, and the fourth things that is happening. Is it clear to all of you the very basic of how fertilization occurs, and from it cleavage, blastulation, and gastrulation occurs? So again, it is a process of cell division, from which the fertilized egg and the fertilized egg is dividing to ultimately form an embryo, which will lead to a fetus. Is it clear? Okay. So I hope this is clear for all of you. I'll just proceed with the next question. Okay. Now, before proceeding to the next question, let's discuss few basic concept of transgenic animals. So, whenever we need a particular strain of choice or a particular plant of choice, till now, what did we do? When we discussed RDT, if we want a particular strain which produces a particular protein, that the gene that we have inserted into it using RDT, we do a selection or a screening so that only our gene, our strain of interest remains, and everybody dies. Okay. And for plants, we do a selective breeding, that we only breed the ones that are giving you good phenotypes. But in case of animals, this this method, in case of animals also, this method was done. But now, with the advancement of technology and with the advancement of science, what is possible is to possibly, what we used to do in E. coli or bacterial or eukaryotic system, that we used to insert a gene within a cell. But that was at cellular level, not at organism level. Here, we are doing the same thing in an organism level. So what happens is, we produce a animal, a entire organism, which is known as the transgenic organism, in which all the cells will have that gene of interest. It's not just one cell, but all cells have. And when these organisms or when these animals propagate, they keep on transforming the information, right? And hence, different novel genes are passed on to descendants. So if I give a small example of it, and so before giving the example, that they are known as transgenic animals. Why? Trans means to transfer, and to genic means we are including or inserting a gene in there. So that is known as transgenic animals. And also, this gene could be from the same species, like from goat to goat, or maybe from somewhere else. Like it could be from a bacteria to a goat, or from a yeast to a goat. That could also happen, or a rat maybe. So if you see this example that I have given a picture of, so this is there's a DNA, then there's a promoter region, and then there's a structural gene. What gene is it? It is a rat somatotropin. Now somatotropin is a growth hormone. So on when this gene is transcribed and translated into the somatotropin hormone, it leads to growth. So what happens is, if I am inserting this particular gene in a normal mouse, then it becomes a transgenic mouse, and it becomes a big and fat mouse. Why? Because in this mouse or rat, the somatotropin hormone is being secreted. Maybe by all cells, and it's not just always all cells that will have this gene. It could be that we have specifically directed the gene towards a tissue or a cell, that okay, these tissues should start producing this particular growth hormone. So if I give you some very small examples, like suppose I want to increase, or I want to increase the bulk production of milk from cow. So I give the cow, give her a particular gene, which will ultimately lead to the production of more milk. So it's like conjugating something with casein, but we have to specifically target it towards the mammary glands, the cell on the mammary gland, so that milk is secreted only from there. We cannot just target it towards hand or leg, like it will be a disaster. You'll not get your targeted product. Similarly, suppose in case of goat, we want or sheep, we want a particular kind of fur from it. So we inject it with the best quality fur, and it's corresponding. We inject it with a gene that will produce the best quality fur. And it should be on the skin, not in any place else. So that is how we do it. Directed can also be done, or in general, suppose not an animal, but maybe we are targeting a plant in its fruit. We want a genetically modified fruit, which will actually give us a therapeutic benefit. It will be beneficial in a therapeutic manner. So in that case also, what do? We direct it towards the fruit, like that. Now, theoretically speaking, every cell should have it, but it's not always that every cell will have. Okay. And let me just show you. Let's discuss some more. Yes. So transgenic, it's like inserting genes within animal which become a permanent process, and it leads to a transgenic animal, and it keeps on propagating. So because genes are also inserted in other organisms, so it's transgene, and transgene means it's engineered and insert. So the different kinds of cells are somatic cells and germline cells. So if you are targeting a particular gene towards this germline, then for sure the next generations will also have those particular targets, and it's inherited. Okay, now let's see how is it actually done. Hmm. Yes, how is this in reality done? So there are many methods. Okay, here we will be discussing on a specific method. So till now we knew that our gene of interest, when we are trying to introduce it within a particular cell, like a eukaryotic cell or be it a bacterial cell, we just transfer it through a plasmid. But here, what do? We do a process called known as a nuclear microinjection. That is, we directly inject the DNA into the nucleus. Now, what is the first step? First step is the transgene or the gene of interest is injected into fertilized egg cells. Just after fertilization, the eggs that contain the original female nucleus plus the male nucleus from the successful sperm. These two pronuclei will soon fuse together. Before this happens, the DNA is injected and uh, into the male pronucleus, which is larger and therefore better target for microinjection. Nuclear is a requires specialized equipment in great scale. So the thing is that we have to transfer this particular transgene to a fertilized cell. So what happens is the male also has a nucleus and the female also has a pronucleus. But the nucleus in male is slightly bigger than the female. So it gets more space. So we generally try to inject it inside the male nucleus, and then that particular transgene containing nuclei uh, that has already like fertilized it is inside the egg, but the nuclei of both have not fused together to form the ultimate embryo, right? So before that it fuses, we are injecting that transgene within it. Okay, and you see, like this, it's a very beautiful process under the microscope. It's very laborious and it takes a lot of time, and the chances of success are also very less, but it's very beautiful. You will see a clear, a very transparent cell, and there's a needle like this. It goes inside and it injects, and that DNA, that is that injection, whatever you're injecting, that is also visible. So it's a highly precise and a very good method. Okay, and then what happens? They fuse and they form clusters. They start dividing the processes that we said, that morula, gastrula, and gastrula. And finally, it is, and now you have cells dividing cell. Now this is a blastula stage. Okay. Now the cell is grown till the blastula stage. Why? Because blastula can be easily implanted, and then that blastula is implanted into a foster mother. So now what is a foster mother? We have known the term surrogacy, right? So here also the term surrogacy comes. Actually, it comes from here only. That what happens is we prepare a female rat by giving it hormones and putting it in an optimum condition so that implantation is easy. So we prepare that uterus, the uterus of the rat, by injections, by hormonal treatments and different other processes, so that it is ready for implantation. And as soon as we are implanting this blastula containing transgene cells into it, it implants and it gets pregnant, and that is how stable integration occurs. And you see its progeny also has the same uh, genes or the same transgene. And some cases it's a failure, it fails to integrate or implant. So that is how it happens. Okay. So what has done? So the now let's just go through the process once again. So till this is it is clear that the nucleus is injected, the male nucleus is injected with the trans. I would like to just stop and ask, is it clear or do you need it in more simple language? Can anyone just respond that if it's clear, or maybe we should go more slower? Okay, let me just remove this picture and go to this. Yeah. So I guess this is much easier. So there's an egg and there's a sperm, and then it forms a zygote. And then there's morula blastula. Now I said the blastula is a good source of ICM and embryonic stem cells, and the gastrula. So that will come later. Why? And how is it done? How is this process actually done? So you see, there's a male nucleus and there's a female nucleus. And we immediately inject it with it before fusion. And you see the zygote. The zygote has the transgene. Okay. Hmm. And now you implant it or inject it into, you implant the blastocele or the blastula stage embryo into the mice, and it will give rise to progeny with the two transgenic progeny. So what happens is the egg is kept in culture during the first few divisions of the embryonic development. Engineered embryos that is the ones which have already the transgene within it, are implanted into the womb of a female animal, which is the foster mother. And then what happens? Then they developed into embryos. And if all goes well, the newborn animals are transgenic. Now, some of the babies will have transgene stably integrated and in the chromosomes, while other the process fails and the transgene is lost. Those that receive the transgene and maintain stably are called founder animals. Why? Because they are the founders of a new kind of population. So they are the founders. And a male and female, when they are mated together, they form a new line of animals carrying two copies of the transgene. Note that the founder animal contains only one single copy of transgene on a chromosome and a heterozygous for the transgene. So if I try to solve this, or we just discuss it in this way, I guess it will be easier. So there's the male and the female. Both of them have one transgene copies that you see here is in red and black, and then they join to form a founder animal which has just a one copy. Okay, now the trans and if two such founder animals which have one copy of each of the transgene, then there's two n copy, and you see 25% of them will be homozygous and rest will be hetero. They have single copies. They see 25%, one out of four. This is the ones which are homozygous. And rest you see. Okay. That is what is happening here. How this 25%, how like the percentage of transgenic animals that we will have. Hmm. Okay. A little more like how is it exactly done? Like now let's discuss what is happening. So first we insert the DNA using micro injection process in an embryonic stem cell from the agar. Now, how is it when we are talking like how is it done? As what did I mention that the embryonic stem cells are very important. Why? Because first and foremost, they are pluripotency. They can form into any particular cell type. They can differentiate and form. So what is done here is one process was to directly inject the cells, the gene into the male nucleus. Here, there's one more process, and this is also used very widely. They take the embryonic stem cells that they grow a particular mice. They subject the mice to proper mouse to high levels of FSH and different growth hormones, and so that the eggs are produced in maximum. They carry the eggs, fertilize it, and after fertilizing it, in the blastula stage, they take the embryonic stem cells out of it. In this embryonic stem cell, they are injecting the transgene. You get it? Before it was for the male nucleus, and now they're directly injecting the transgene into the embryo stem cell. Now, what will happen is, now when you're injecting into the embryonic stem cell, there will be homologous recombination, because obviously the embryonic stem cell has like normal genetic makeup, and then you are doing that. And these stem cells are inserted back into a blastocyst or in the blastula stage, right? So that they grow and they finally implant. And again, this implant is not within the same mice. It is in a pseudo pregnant female mice. Okay. And that pseudo pregnant female mice will now like host the embryo. It will become a fetus and ultimately lead to offsprings. And you see the offsprings are, these are the transgenic offsprings. Okay. Hmm. Is it clear that what is happening? Let me just, yeah. Hmm. So what happens is there's a host blastula. I'll just repeat the same process once again. So there's this host blastula. So what does this host blastula do? It has ICM. From that ICM, that is inner cell mass, these are extracted, the embryonic stem cells, and we introduce the genes into it, and then we let it grow. Let it grow means we insert it back to a blastocell, but in a different blastocyst and not in within that same parent. Again, after the blastocyst grows and it is in proper form and in optimum condition for implantation, we put it inside a foster or a surrogate mother or a pseudo pregnant mother. And then the embryo develops and it becomes a chimera. Now, do any one of you know what a chimera is? Any idea what is a chimera and why? Now, when we are inserting the gene of incest into the ASCs, the embryonic stem cells, and we're implanting it or we are putting it back into a blastocyst, that blastocyst origin is from a different embryo. And then again, we are putting this in a foster, within a foster mother. So that makes it two different embryos from different origins that we are using. One was before gene introduction, and second was when we have reintroduced inside a blastocyst, and then we are implanting into a mother. So this is two different sources of. If I just try to mark it, one source is this, and one source is this. So we have two different sources of embryos. So what will happen is they'll form a chimera. Now a chimera means it's a mixture. It will not have pure, like just all cells with gene of interest. No, some of them will have, and some of them will not have. So if we look into more things, what happens is, yes, no, there's no more thing. This is the ideal thing that is happening. Yes. So is this process of embryonic stem cell or how animal biotech, this is it clear to all of you? Animal biotechnology or additive using animals, transgenic animals, is it clear? What is the use of embryonic stem cells? Okay, Amardeep says yes, so I expect. Okay, so that is now let's go to the next question. So we'll understand why did I brief on all of this. Okay, let's try to solve this question. So what type of cell combination should you start with? Sorry, I have a chat. Acha, what is agouti mouse? So agouti mouse is basically a mouse with a brown fur. So that is a agouti mouse. It's name of a strain or name of a species of mouse which has a different kind of brown fur to it. That is a agouti mouse. If I'm not very wrong. So let's try to. It's not a mouse. It's a rat. I'm sorry. I keep on saying mouse, but it's actually a rat. I guess. Yes. Okay. So rat and mouse are different, and here it is written mouse, but the first one was a rat, and this is a mouse. Okay. So let's now again we have a cloning experiment with the rat. Now let's try to solve this. What type of cell combinations would you start with if you wish to clone an animal, for example, a lab rat? Select the right response. Anyone? Amardeep, it seems it's clear to you, so you should just try it and answer. Just try. Let's go through all the options. Like try to analyze that what these options are and what it means. Try to think of the first scenario, this one, where we were trying to inject into the micronuclei. So this I will repeat this once again. I guess this is new to all of you. So what we used to do is that we used to inject a transgene within the male nuclei so that it fuses with the female nuclei and forms a zygote, and that zygote divides and has the gene of interest in all cells. And what is the second scenario? What can it be done? That there is the sperm and the egg, and we let it first divide to form zygote. So after one round of division, then we are trying to integrate or insert the gene of interest. So what will happen is few cells will have the genes and few cells won't have the gene. So it will form a mosaic of it. So that is how all cells will not be transgenic, but few of them will be. So if you keep this in mind, what could be the possible answer? Yes. So what could be the possible answer for this? That if we are trying to, what type of cell combination should we start with to clone an animal, to wish to clone an animal? Right one. So you need a donor, of course, and a recipient. Donor is one where you will inject the transgene, and a recipient is one with what it will fuse to form the zygote. So what could be? Just have a look at the picture in your left, and maybe you can try to answer this. Okay. So I guess this concept is not much clear to all of you. We have two participants, and Arushi, is it clear to you that, well, is it okay for you, or you're not understanding it? You can be frank with me. For this one, it will be embryonic stem cells, which are the donor nucleus, stem cell for donor nucleus, and female rats will be the eggs will be the. Oh, okay, I understood, little ma'am. Okay. I will just repeat this entire thing for you once again. Female rat eggs will be the recipient, right? So it will be male nucleus because we are going to inject in the male nucleus, and then the recipient would be female. Okay. Arushi, let's go through the entire thing once again. Okay. Okay. So what are we discussing? Here we are discussing embryonic stem cells. Okay. So what are these embryonic stem cells? Suppose this is your egg and this is your sperm. It will fuse to form a zygote, right? Now, this zygote will have many different stages. The first stage is the morula. From there, it will be blastu blastula, and then the gastrule. Okay, which forms the ectoderm, endoderm, and that like that. I'm not drawing it at part the scale. Now, this blastula is very important. Why? It looks somewhat like this. These are the trophoblasts, and here is the inner cell mass, ICM. Why is it important? Now, this ICM has embryonic stem cells. Now, these embryonic stem cells have this property of self renewal. We all know it's pluripotent and it is pluripotent. So, it is actually capable of forming any kind of cell. Till this much is clear, Arushi? Is it clear till this much? Okay. Now, whenever we are trying, now you understand these ESCs are very early stage of embryonic development. So when we are trying to manipulate a particular gene or any organism so that it is able to produce something of our interest, or it can become something that we want to, right? Then we need to exploit it in its early embryonic stages, in a part where if you're exploiting something and it's going to be sure shot that okay, this gene will be changed, this particular product will be produced, this will be done. So similarly, the ESC is such a stage of cell which is highly exploitable. And also what? Because it's within the blastula, so blastula is a stage of embryonic development which is implanted in the uterus. Okay. So if we are putting anything inside ESC, it will actually lead to an organism which will sure shot have the modified gene in it, because this is a very early stage of embryonic development. Is this much clear, Arushi? Okay. Now how we do it? How is this process done? So there are many methods. So how embryonic stem cells are actually used, if you want to discuss. Hmm. So this is my embryonic stem cell. Okay. I put a transgene within it, which is so now my embryonic stem cell has this transgene in it, and I place this embryonic stem cell in the blastula or the blastocyst. The cell, I mean the entire cell that has the different embryonic cells. So if I implant it within it, it will form different, it will divide and it will form all the cells or few cells which will have this gene. And this will ultimately lead to a transgenic animal which will have few genes which were like inserted in its ESCs. Okay. Now this process is not very clear, very not clear. I mean by it's not a very straightforward. Okay. Arushi, I'll share my screen and we'll just begin. Okay. Just let me know if my screen is visible. Is it
Visible,
visible?
ma'am.
Okay, 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 okay. Cool. Okay. So now this process is not very straightforward. Why? Because this is the embryonic stem cell. This is the embryonic stem cell plus the transgene. Okay. Now this embryonic stem cell is taken from suppose source 1. Okay. And it's grown with the transgene is inserted and the blastocyst is grown to till this, and then what we do is we implant this in another organism in which sorry, we not here, we grow this in another organism which is source 2, right? And within this source 2 this growth happens. Hmm. We grow the cells in source 2, and this source 2 is nothing but the cells which are growing. Once this have grown, now what do you mean by source 2? That now this is one ESC, but it is capable of growing within a blastocyst or a blastocele. So we have to transfer it into a new blastocele, which is source 2, where this will grow and form like this. Now the blastula stage is ready. We have to implant it in a foster mother, which is source 3. So this use of 3 different organisms. One gives the ESC, one harbors or lets the blastula grow, and the other one actually implants it within its womb. And from here you have your transgenic animals. Is it clear?
Yes, ma'am.
Now is it clear? So will you be able to now understand this question, that what should be the criteria? If you try to recapitulate slightly, that what are the cell combinations that you have to start with if you wish to clone an animal?
I could not hear you properly.
I mean, can you just try to answer this question? Like, what should be the combination of cells that you should use if you want to wish to clone an animal? Early embryonic cells for donor nucleus. There is where you will donor nucleus means somewhere you will actually inject the transgene and it will donate it to some other cell. And then you are letting it grow in a blastocele in the next one. So it's the female rat.
Ma'am, I
Yes.
mean, embryonic cell only will be inserting the transgene, ma'am.
Yes, in embryonic stem cell, you will be
Okay.
inserting. Yes. Hmm. Wait. See. This is the your DNA construct, okay? And you are injecting this DNA into an embryonic cell, which is from a agouti mouse. You see, within here, the recombination process is happening, and you let it recombine. And then what you do, you insert this stem cell into an early embryo of a different mice, which is maybe from you take the blasto. You actually we don't insert it. We take the we let the blastocele grow in that mice also, and we take it out, and in that we are inserting the transgenic embryonic stem cell. Is this part clear, or is it sounds a bit confusing?
Clear, ma'am.
Clear, right? So this is a different source. It's a white male mouse from there. Okay. Blastocyst. Okay. Then you are implanting into a proper mice, a female mice. There what happens? It will grow within that. You're implanting it. Till now you were inserting, you were putting it, you were taking out, you're doing all sort of these things. But now you're actually implanting it so that it gets pregnant and it produces the offspring which has the transgenic gene in it. Okay,
Okay, ma'am.
clear? I would suggest you go back, uh, read this slightly, and it will be clear to you. And then if you still have any doubt, do post it in the discussion forum. I'll take this topic again in the next week. Okay. Maybe we will just discuss it and not any in question answer format. Okay. Okay. There's one more question on it. Let's try to solve it. So in a laboratory, you're preparing for cloning experiment that will necessitate the use of embryonic stem cells. Choose the correct combination of embryonic stem cell phases. Option 1. Try this and let's discuss. Uh, do you think this answer is correct? Amardeep and Arushi, do you feel this answer is correct, or it could be one of four also? I was sure option 2 was correct. Okay. I was sure option 2 was correct. You mean 1, 2, and 3? Option 2 means? Oh, you mean 2 is definitely a choice, but about the second choice, you are not clear. Yes. Okay. Yeah, so this is very clear. The first option, they're unsuitable for cloning studies. Definitely not. They are the ones that are used for cloning. No other cells are that much used. They can be easily extracted from blastula stage. So yes, that is why we aim for embryonic stem cells, because it's available in the blastula stage itself. And also they can multiply endlessly in the due to its self-renewal and pluripotent self-renewal capabilities. It is like it can multiply endlessly in the specific cell culture that we are trying to grow it in, and they are highly specialized cells. No, this is very vague, so this will not be done. Okay. Slightly talking about cancer once again. So the question states that tumors are assumed to be benign in their early stages until a small number of cells escape the tumor origin and over time develop secondary tumors at various sites and organs of the body via a highly specialized process. What do you call the process? And also figure out what attribute does not constitute a malignant tumor. This is answerable. So I would like a discussion on this. Anyone? Okay. So let's first discuss what metastasis is, and then we can like come to the answer. So suppose you have a primary tumor formation at a particular site. This is how the tumor forms, you see. Then it's a localized invasion. Then happens a intravasation, that is, it uh, interacts with the adjacent platelets, lymphocytes, and other blood components. So what happens is it interacts and then it's transported through circulation to other parts, which could be organ, which could be bone, which could be even the brain. And there extravasation occurs. That is, it comes out for some micrometastasis and ultimately it colonizes to form a macrometastasis. So that is how a primary tumor formation can lead to a secondary tumor and ultimately it forms a new tumor in a different way. So if I just read out what is written in the legend, figure legend, the initial step is localization or invasiveness, enabling in situ carcinoma breach the basal membrane. So in this step, I was saying if this is the basal membrane and here are the tumor cells, normal cell, it has to reach out to become cancerous. So now, if these cells were already cancerous, again to go outside, it has to breach this membrane and then only go out. So that is locally invasion. Then what do they do? They travel, they intravasate through the lymphatic or the blood microvessels. That blood is the lymphocytes, blood. These are the carriers of it that takes it to other places. So they transport then they transport the cells through generalized circulation to anatomical sites where they could be trapped, and subsequently they penetrate and form dormant environments, microenvironments, where again a tumor formation begins. And finally, when they acquire the ability to form or colonize there or to form colonies, they form a new tumor. And that is what is the last step. And uh, that is how the macrometastasis happens. But keep one thing in mind, this process doesn't seem very easy. Like you see, many people generate like metastasis occurs after a long time. It takes time. It's not that always it will happen very fast. It is eventually is a long process. Doesn't happen overnight. And it has to follow or fulfill all this step by step, all these processes, finally to again form a tumor in the another region. So the small probability of successfully completing all steps of this cascade explains how likely that any single cancer cell living a primary tumor will succeed in becoming the founder of a distant macroscopic metastasis. So now let me come back to the question that what is this process called? It's metastasis, and also figure out what attributes does not constitute a malignant tumor. So let's see the attributes. Acceleration of cell cycle, that cell cycle happens very fast. Genomic alteration, genetic mutations, invasive growth, we know that. Increased mobility, it makes the cells mobile. Chemotaxis, that is, it senses, it produces, secretes something, and that is sensed by the other adjacent cells, and that is how it crosstalks with its neighbors. And it changes the cellular surface, the morphology of the cancer cells change, and finally, it's secretion of lytic factors. So that comes to the answer. It is metastasis and pluripotency. So is it clear?
Yes, ma'am.
Okay. Okay. Now, the last question of today. Now, are you aware of Hardy-Weinberg equilibrium, or do you know how to like solve sums of this? Are you aware of the formula that is used to? Okay. Are you? No. Okay. 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 So the concept is that in a population, whatever might be the recessive or homozygous or maybe dominant, whatever might be the genetic makeup, allele frequency remains same. So what do you mean by allele frequency now? Now, what is allele? It is a variant of genes, right? Alleles. So what happens is the position where the allele is one or human or one chromosome in a particular individual, the frequency of how many individuals have that particular allele in that same position. That is the allele frequency. And that actually makes the genotypic constituent or the makeup of a particular individual. So the formula looks something like this. P square plus 2pq plus q square equals to 1. 1 why? Because it's stable. P square is actually P and P plus 2pq. Q and Q equals to 1. So these are alleles. P and Q are the alleles. Okay. Here it's G and G. Well, small g and capital G. So what does this question say? Let's see. Individuals with wild type gene, G in human, make a glycoprotein. So small g is the wild type. The mutant version of the glycoprotein works as a receptor for the seasonal flu virus, allowing it to enter the host cells. As a result, person with mutant gene becomes ill. Now, capital G is the mutant. A dominant allele produces the mutant glycoprotein. So this is again dominant. That is why we have written it in capital also. If 84% of the population is vulnerable to seasonal flu, individuals with GG and G, right, they are vulnerable. What is the frequency of the mutant allele? That means that if 84% of the population is vulnerable to the flu, that is the person which has the mutant allele, if they are vulnerable to it, so what is the frequency that the mutant allele is in the population? That is how we find that what is the frequency. I said allele frequency remains the same, that it is conserved. So how do we do that? So now let's do it in that way, that GG. Okay, GG and small g small g. This one is 84% plus g square equals to 1. So small g equals to 1 minus 0.84. Simple mathematics, right? So when we subtract 1 minus 0.84, it becomes 0.16. So g is equals to 0.4. Now G was the wild type. We have to find the frequency of the mutant in the population. So if G is 0.4 and we know that G plus G equals to 1. So 1 minus 0.4 is equals to 0.6. Clear how this is done? Was it clear to all of you?
Yes, ma'am.
So this is how generally you get sums, like sometimes they will just give you number of like X 100, the total population size is 200 and 100 people have PP. So what is the frequency of P? So you just divide it and you get the frequency. That is how these sums keep on like repeating in that way, not always like it's the same. So is it clear as of today that how these are done? And do you want any other discussion on any other question? Hmm? Anyone?
No, no, ma'am.
I haven't missed any question. Okay. Amardeep, you need anything? Any help? How many participants do we still have? Okay. No, ma'am. Okay. So thank you for joining. We'll meet again next Friday, same time. And if you have any questions, as I've mentioned, please do post it in the forum so that we can be able to address it uh, in the next class, or in the whoever is going to take which. So we can do that. And thank you for joining. Thank you all.
Thank you.